other genetics too, like this is the project is just catalog. It's a catalog of things. It's not defining whether this difference is responsible for some disease or not. But what other scientists are doing is they are seeing whether this difference means that this person B has cancer or not. Now, if I compare all these 5 individuals and I find that having this difference in B means B gets cancer and rest of them don't, then I can hypothesize and say having this difference leads to cancer or increases the risk to cancer. We don't ever say he'll get cancer. Whenever we talk about in a population level, we say the risk of, like whenever you read news, like the risk of heart disease or risk of diabetes, it's never that you'll get diabetes or you'll get heart disease. It's always a risk, because smoking increases the risk of cancer. It's not that smoking not always causes cancer. There are people who smoke all their lives, 30 packs, like 30 cigarettes a day, and they are all fine. And some people get lung cancer by just passive smoke. So smoking by itself increases the risk. It's just that whether you get a cancer or not is dependent on various other factors. So what we say is, oh yeah, having this difference here means the B has a higher risk of having cancer. So like if you look at this triangle, this is a typical triangle kind of diagram that people make for defining relationship between whether that particular variation has some significance or not. And these black lines that you're seeing, like the triangles you're seeing, that means all the individuals that have this variant, say for example from here to here, these combination of variants are associated with some sort of a disease. So this gives you a measure of association of particular variant to a particular disease. Now for each disease this diagram would vary obviously, and so this is how HapMap project is like. This is defining the variation among populations. And so people have, like, this is the whole of the human genome. This is updated till December 2009. So if you look across the genome, these are all the chromosome 1 to chromosome X and Y. So every chromosome has these circles actually denote various diseases. So if you read, this is acute lymphatic leukemia, and like alcohol dependence even is a disorder, and asthma, dermatitis, esophageal cancer. So all of these, like type 1, type 2 diabetes, stroke, pulmonary function, and like obesity is a complex disease. Like these are all disorders which are not defined by one particular gene, because obesity or something like that is a multigenic. So like a lot of genes are involved. So all these are various disorders, and here's sort of an expanded list. So you can, if you read through it, you'll sort of see that many of them are type 2 diabetes, obesity, inflammatory bowel disease, various cancers, arthritis, various coronary disease, asthma, glaucoma, height, hair color, eye color. Even though Krishanu was saying blue eye color is Mendelian, it's not actually Mendelian. And even like something like freckles, which is like more, if you see like white skinned people have freckles. When they are exposed to sun, they get freckles, and that also is multigenic. And what is your HIV viral? So even AIDS, even though AIDS is caused by virus, HIV, the severity of the disease is something that is dependent on the human being which is getting infected. Some human beings get very severe infections so fast that they die very fast. Some human beings can survive longer, and given an antiviral therapy, they probably are living a normal life. So even this, which sort of is a disease which is 99% fatal, has terms in terms of what would be the final outcome. So you see this term GWAS, it's sort of is a very hot term these days, where lot of populations are being studied. Even like we haven't started this in India as of now, but lot of Caucasian populations and African populations are being used to find causes, that variation that would cause increased risks for various diseases, like here listed here. So this gave you an overview of what human variation is about. But the problem with studying humans is that it's post facto. You can only study what is present. You can't go back in the history. You can't do any kind of genetics with human beings because it's ethically impossible to do. So what we do is, with humans we study as the data is presented, as it is, kind of like as it is basis kind of study. So we have to use model organisms when we want to try to understand what are the genetic basis of these kind of variations. And these variations are actually quantitative. So the reason it's called quantitative is because they differ from Mendelian. So Mendelian traits, if we plot Mendelian traits in a population, there are only two variants, a capital A and a small a. Say for example, this is the dominant phenotype. You know dominance and recessive. Dominant is like the phenotype that presents itself, and the recessive, which would only happen if the dominant phenotype is not present. So it is like this, and the recessive is like this. So it's a on and off system, either on or off. There's no intermediate. So it's a binary system, on off. Now if you compare this to what's called quantitative traits, they follow a distribution which is called like this, which follows like this. And there are many, like lots of closely spaced lines, and the arrow says that this is 0 and this is 1, and then there are fractions of it. Yeah, so the real example would be in Mendel's flower example. So you have a white flower here and a red flower here. So when you take a white flower plant and cross it with a red flower plant, you either get white or red. You would not get anything like no shades of pink, right? So you have either white flower or red flower. In this case, this is a, say for example, white, and this is red. If he did this with garden pea. Okay, so in garden pea this is the case. Okay, now there is another part called antirrhinum, which is dog flower. Okay, now if you take antirrhinum, you take white flower and a red flower, take white flower plant and cross with a red flower plant, you will get all shades of pink. Now you would not be able to tell how much is white in there, how much is red in there, because they are all varying in small fractions. So for example, another good example is human height for this. Say for example, this is 4 feet and this is 7 feet, right? In a population you will get all kinds of individuals, but majority, say for example, average human height of Indian individual is 5 feet 6 inches, right? So 5 feet 6 inches would be somewhere here, because most of the people are 5 feet 6 inches. Very few people are very short, very few people are very tall, and most of the people lie in the sort of what's called the middle range. And this is so these kind of phenotypes or characteristics are very difficult to understand genetically, because here you know what is the difference. It's an on-off system. You remove this, you get this. You remove this, you get something else. So like there is like you remove a gene, you get a phenotype. Here you remove, even if you remove this, nothing's going to change. Like, okay, you take a, say for example, you've seen like it's a very typical example that probably is very common. Two tall parents would have taller kids, or even sometimes a shorter kid, or two short parents have taller kid. So how does this happen? Like if I were to take two red flowers and cross them, I will always get red flowers, no matter what happens. Okay, but I am taking two short parents and their kids are tall. Like I had like two parents are 5 feet and the kid is like 6 feet. So how does that happen? This is because these kind of characteristics